Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore a truly fascinating topic: sex-influenced and sex-limited characters. Have you ever wondered why certain genetic traits appear so differently in males and females, even when they inherit the exact same DNA? By the end of this video, you will understand exactly how the internal environment of a body can change the rules of genetics. To begin our exploration, let us imagine two people who are given the exact same recipe to bake a cake. One person bakes it in a standard oven, while the other uses a microwave. Even though the instructions are identical, the final cakes will be completely different because the environment changed. In genetics, this environment is often the hormonal difference between males and females. Sex-influenced traits are characteristics where the exact same genetic code produces a different physical result depending on the sex of the body. This matters immensely because it proves genes do not act in a vacuum. The most famous example is male pattern baldness, where a man might lose his hair completely, while a woman with the exact same genes only experiences slight thinning. Building on that analogy, let us dive deeper into the genetic mechanism behind these traits. It is crucial to understand that these are autosomal traits. Because the genes are located on autosomes, which are the non-sex chromosomes, both males and females inherit the exact same two copies of the gene. So why do they look different? The secret lies in the dominance relationship. The presence of specific sex hormones, like testosterone or estrogen, actually reverses or alters which allele acts as dominant. This means that a heterozygous individual, someone with one copy of each allele, will show one phenotype if they are male and a completely different phenotype if they are female. This prevents us from confusing them with sex-linked traits. Now that we understand what sex-influenced characters are, let us look at the mathematical relationship that defines them. Conceptually, we can express this as P equals F of G and S. In this equation, P stands for the final physical appearance, or phenotype, of the organism. The variable G represents the genotype, meaning the specific alleles the individual inherited, such as capital B or lowercase b. The variable S represents the internal sex hormones present, like testosterone or estrogen. Finally, the function F represents the biological process of gene expression itself. What this formula tells us is that if you keep the genotype G constant, but change the hormonal environment S from male to female, the final physical output P will change completely. The exact same genetic input yields a different physical output when the hormonal multiplier shifts. Before we move on, let us see this concept in action with a concrete example. Suppose a non-bald man marries a bald woman. We are asked to find the genotypes of the parents and their children. We chose this standard Mendelian cross to see how the alleles are passed down. First, because the man is non-bald and this trait is recessive in males, he must have two copies of the recessive allele, so his genotype is lowercase b, lowercase b. The woman is bald, which is recessive in females, so she must be capital B, capital B. When they reproduce, each passes exactly one allele to their children. Therefore, 100% of their offspring will inherit the heterozygous genotype, capital B, lowercase b. Physically, this means every child carries the exact same genetic code for this trait. So we have proven that all children have the heterozygous genotype. This brings us to deriving their actual physical appearance. This chunk of our derivation establishes how the exact same genes create different traits. For a son, the internal environment is high in testosterone. This specific chemical environment interacts with the genes, making the capital B allele act as dominant. As a result, the son will be bald. However, for a daughter, the environment is high in estrogen. This causes the dominance to flip, making the lowercase b allele dominant instead. Therefore, the daughter will be non-bald. The mathematical reasoning here shows that the genetic inheritance is identical, but the physical manifestation is opposite. This perfectly illustrates the intuition behind sex-influenced traits, leading us to our next major concept. Now that we have mastered sex-influence traits, let us look at a more extreme scenario: sex-limited characters. Think of a locked treasure chest that both a brother and a sister inherit. They both own the exact same chest, but only the sister was given the key to open it. The brother has the chest, but he can never use what is inside. In genetics, both males and females inherit the genes for a specific trait, but the trait is only ever unlocked and physically expressed in one sex. The other sex carries the genes silently and can pass them on, but will never show the trait themselves. A classic example is milk production. Bulls carry genes for high milk yield and pass them to daughters, but bulls obviously do not produce milk. Understanding this hidden inheritance brings us to the biological rule governing it. A sex-limited character is an autosomal trait that has zero penetrance in the opposite sex. Even though the genes are located on autosomes and inherited equally, anatomical or physiological differences completely prevent one sex from expressing them. The genes are essentially turned off, regardless of whether the individual is homozygous dominant, heterozygous, or homozygous recessive. This is incredibly important for evolutionary biology because it creates extreme sexual dimorphism, where males and females of the same species look or function in radically different ways, such as male peacocks having massive, colorful tail feathers, while females do not. This concept of hidden genes leads us to a formal mathematical expression of sex-limited traits. The formula defines the probability of a trait being expressed. Here, P stands for penetrance, which is simply a probability value ranging from 0 to 1. The variable T represents the genetic trait in question, such as milk production. S1 represents the permitted sex, like a female cow. Given the permitted sex, the probability is 1.0, meaning a 100% chance of expression. Conversely, S2 represents the restricted sex, like a male bull. For them, the probability is exactly 0.0. Physically, this means that no matter how strong the genetic variables are, multiplying by a penetrance of 0 ensures the trait will never physically appear in the restricted sex. Now that we have this probability formula, let us apply it to a real-world example. We are looking at hen feathering in chickens, where the capital H allele is dominant. We are asked to cross two heterozygous chickens and find the offspring ratios. We use a standard monohybrid cross because the genes themselves are inherited normally on autosomes. Each parent has the genotype capital H, lowercase h. During reproduction, each parent has a 50% chance of passing on either allele. When we map out the combinations, we get a classic genetic ratio. 1/4 of the chicks will be capital H, 1/2 will be capital H, lowercase h, and 1/4 will be lowercase h. This final answer gives us the genetic blueprint, which we must now interpret physically. With our genetic ratio established, we now derive the physical phenotypes for each sex. This chunk of the derivation applies our rule of zero penetrance. First, we look at the female chicks. Because cock feathering is strictly limited to males, the trait is completely suppressed in females. Mathematically, Their penetrance for cock feathering is 0. Therefore, regardless of whether their genotype is capital H, capital H lowercase h, or lowercase h, 100% of females will be hen-feathered. Next, we evaluate the males. Since they are the permitted sex, we read their genes normally. The capital H is dominant, so males with capital H or capital H lowercase h will be hen-feathered. Only the males with the homozygous recessive lowercase h genotype will display the flashy cock feathers. This proves how the physical manifestation is blocked in one sex. Now that we have calculated examples for both concepts, let us look at this diagram to visualize how they compare. You are looking at a Venn diagram that beautifully summarizes our entire lesson. On the left, the circle for sex-influenced traits highlights that these are expressed in both sexes, but the dominance reverses, just like we saw with pattern baldness. On the right, the circle for sex-limited traits shows that these are expressed in only one sex due to 0 penetrance, like milk production in cows. But the most important part of this diagram is the intersection in the middle. It teaches us that despite their differences, both types of traits share a common foundation. They are both caused by autosomal genes, not sex chromosomes, and their expression is heavily controlled by the internal hormonal environment. This brings us to the end of our journey, so let us summarize the key takeaways. First, always remember that both sex-influenced and sex-limited traits are driven by autosomal genes, meaning they are inherited equally by males and females. Second, the difference lies in expression. Sex-influenced traits flip their dominance rules based on whether the body is male or female. Third, sex-limited traits take this a step further with an absolute restriction, completely turning off the trait in one sex. You have now mastered how the internal environment of the body determines how a gene is actually read. Excellent work today, and keep exploring the fascinating world of genetics.